SAD and MAD studies are used in early phase 1 development to understand how a new drug behaves in humans. SAD stands for single ascending dose. In a SAD study, participants receive one dose of the investigational drug, usually starting at a low dose that has been selected from the non-clinical package. The aim is to build an initial picture of safety, tolerability, and PK. This describes how the drug is absorbed, distributed, metabolized, and cleared from the body. MAD stands for multiple ascending dose. A MAD study looks at repeated dosing. It helps teams understand whether exposure builds up, how it changes over time, when steady state may be reached, and whether tolerability looks different after several doses. The two designs are often linked because the learning carries from one stage to the next. Results from SAD can shape dose levels, sampling schedules, and review points in MAD. Some programs use a combined protocol for that reason. This can support a more efficient early development plan, so long as the design is tightly controlled. Dose escalation needs clear rules before dosing begins. The starting dose should be justified, the rules for dose increases should be usable, and the criteria for moving between cohorts should be defined before the study begins. Each step depends on the emerging data, including safety findings, PK results, and whether observed exposure matches expectations. In early cohorts, sentinel dosing is often used. This means a small number of participants are dosed first and monitored closely before the rest of the cohort proceeds. This gives the team an early safety check before broader dosing within that cohort. Data timing can shape whether escalation decisions are reliable. Safety labs, ECGs, adverse event data, and PK results need to be available when dose decisions are made. If PK data is delayed, the team may be forced to decide without the full picture. If early results show that the drug peaks later than expected or clears more slowly, the sampling schedule may need to be reconsidered so that later cohorts capture the profile properly. Repeated dosing can reveal patterns that a single dose cannot. A drug that appears tolerable after one dose may behave differently once participants receive it over several days. MAD can also support early understanding of pharmacodynamic markers, where a suitable marker has been selected and measured in a controlled way. These results can inform later dose planning. MAD can support thinking about dose level, dosing schedule, and whether the intended regimen is likely to be workable in a patient study. This gives a more grounded view of repeat dose behaviour. Optional elements, such as food effect work, can be useful when there is a clear reason to include them. A food effect arm may make sense if meals could affect absorption, or if likely, clinical use makes that question important early. But the risk comes when flexibility becomes too open-ended. The protocol should specify what can change, why and how decisions will be made. Execution matters because these studies often run on compressed timelines. Dose administration windows, sample timing, safety checks, and review meetings all need close coordination. If it isn't, the resulting data can be harder to interpret. SAD gives the first human read on exposure and tolerability, while MAD builds on that by examining repeat dosing, accumulation, and longer patterns of tolerability. Across both, the study is only as useful as the data the team can generate and the decisions they can make from it. To learn how Quanticate can support your next trial, email inquiries@quanticate.com or request a consultation through our website. Thanks for watching, and don't forget to subscribe for more practical insights on clinical trials and modern drug development.